All right, good morning, everybody. Hope all is well. Your exam is not until Friday, so we're going we're going to review. Uh, while I was checking the worksheets, you need a little review on cell transport, so we're going to do a Nearpod this morning. And we're going to do it live. We're not going to do self-paced. Everybody needs to pay attention. We'll do this one together. Some of y'all been tipping out on me. All right, so the link is in the chat. As soon as possible. All right, so this is a video, but it has some question areas to it. So I'll know whether you participated or not, whether your contribution to the questions. Excuse me. Give it one more minute for folks. I think one or two people are still missing. Go ahead and log in, sign up. Here's one. Okay. Good morning. Good morning. Good morning. I'll put the link in the chat again. Okay. And we're doing this together. We're doing this together. So everybody should be logged in by now. There's a way for me to check. Okay. Let me see. Right, Avenay, okay, hurry up and log in. We'll wait on you. I should see everybody's name. All right. Says, what do you already know? So don't say nothing because you know something, but shed a little bit, you know. I'm gonna stop sharing for just a minute while you answer that question. And again, this is on your test is Friday. It's multiple choice. It's about 35 questions. Um, and one of the things I noticed when I was grading the worksheets on cell transport is that there's a little bit of confusion there. So I need you to participate. So I'm waiting for everybody to put an answer right now. I got three people who have answered the question. What do you already know? This is an interactive. So while you're doing this with me, I will pause for a few minutes. All right. Even if you just say what Raymond said, there are two types you need to know. There are two types. Alicia, I don't see you logged in. I need to see everybody's name on the list. This is participation. You can say more than two types. You need to say what they are, active and passive transport. Oh, goodness. See what I mean? Trying to help you out here. Trying to help you out. Still waiting on. Thank you. All right. Got my other three people in now. I need an answer. And if you don't know, you should be looking it up. Like I said, this is part of our review for Friday. You get a little, you're lucky you get extra time. The other class took their test yesterday. Trying to help you, trying to help you. I might put some extra questions on the test. All right. I got two folks who still haven't answered, but we're going to move on. If you choose not to answer, that's on you. Thank you. One more. Andre, are you with us? All right, let me go back to sharing. You, well, we're going to know more. By the time we finish, you're going to know everything you need to know. And if you don't, you need to ask questions.
You ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this.
Okay, I'm going to back that up just a little bit. Whoops. All right, answer. Hold on. Maybe I shouldn't have did that. Anyway, the word I was trying to get y'all to see was homeostasis. That's a vocabulary word. You need to know that that means maintaining a stable environment, a steady state environment is what they call it sometimes. I am not going to mess with it like that again. I look like I just messed it all up.
Could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer.
Okay. All right, I need answers to the question. Answers to the question. I want to see 100% participation. Right now, I got 50% of y'all not answering yet. Wake up, folks. Let's not hold this up any longer than it has to. Everybody needs to participate. You logged in, you might as well participate. Every time you see a blue dot, that's where she's going to stop and ask you a question. So just be ready. All right, and most of you were paying attention because the last thing she said was the right answer to the question was the cell membrane is made up of phospholipid layer. Again, if you're looking at your macromolecules chart, we looked at the fact that the fatty acid monomer is also called a phospholipid layer molecule because it's got a phosphate molecule with the long strings of lipids hanging from it. All right. See, the same one person not participating.
Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and
Okay, I'm going to try stop it whenever possible. So again, polar means just think of the North and South Pole, or think more like a magnet. You're familiar with the fact that the magnet has a North and South Pole. Okay, and opposites attract, and poles repel each other. Okay, so there is a positive end of the molecule and a negative end of the molecule.
they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport.
Okay, simple diffusion is what now? Like I said, these questions are always to see if you're awake and paying attention because they always say the answer right before the question. 80% of you are paying attention. All right, the other two people are not answering. All right, but the 80% of you who did answer, you are correct. It is passive transport. Thank you, one person. No, it is not active, it is passive. And again, what she said was passive doesn't require energy. It flows through without something forcing it to do so. The membrane has pores in it, big enough for these molecules just to pass right on through.
Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low.
Okay, facilitated diffusion. The molecules move from what concentration to what concentration? Again, this is also referred to as the concentration gradient. And that refers to the flow of molecules. Which way are they going? Come on, folks, pay attention, stay with us. Thank you to the 80% of you who are actually actively following. We don't have to keep waking you up. All right, so yes, it's going from high to low. Okay, remember I gave the example of being in a crowded room, and as soon as the doors open to the empty room, you just want to run out because you want space. Molecules are like that. They like space. So if they're in an area where they're all squished together, they're going to move to the area where there is less crowding.
It doesn't require energy, though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all
Whoops. All right, that was one of the questions that people were getting wrong. That glucose and that molecule, one of the circles on the worksheet was about glucose. So it was simple diffusion, but it was facilitated diffusion. So just remember that it was diffusion, it wasn't active transport because it was glucose, but it was facilitated diffusion in that the molecules are too big to go through the regular pores, but they can get through the specialized protein transport molecules.
are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose, but what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We would still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates. And when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself.
Okay, what powers active transport? It is called the energy molecule. Okay. ATP is the correct answer. And just remember that ATP is the name of the energy molecule in the cell. There's questions about that on the test. So remember what ATP is. It actually stands for adenosine triphosphate. So it's a little phosphate with other things attached to it. Or 3 phosphates, actually triphosphate with an adenosine molecule attached to it. So when it detaches, it can be diphosphate or monophosphate as it moves along.
One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside. And this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids.
All right, which one allows you to take in fluids? And again, these are vocabulary words: phagocytosis, exocytosis, pinocytosis, and the endocytosis that she mentioned earlier. Those are vocabulary words that you need to know. And yes, it is pinocytosis.
So, to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit. They sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. I mean, all cells have membranes, but not all cells have a wall. But plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there.
All right. Okay, I'm going to stop sharing while you answer this question. You can also answer this as to what did you learn just now that you didn't remember before? Hmm. Oh, that's interesting. Imani, that's an interesting analogy as well. I'm learning that analogy with the oil and the gas and the motor and all these wonderful things that make the car go or not go. All right, still waiting on a couple of people to answer. Okay, everybody. But I noticed that nobody has noticed. You know what should be amazing about this? This is going on inside you right now. You had breakfast this morning, and your cells are operating this active and passive transport right now. And that's what's keeping you going. All those little ATP molecules are working with you right now. Some of you, the ATP is a little low, but they still working. They still working. All right. All right. They just said bye. All right. Um, let me see. Let me stop this. Get out of this. And we're leaving. We're leaving because we're done. Okay. They ask you so many questions. All right, we're going to look at macromolecules now. Let's just keep on going since today is our review day. Trying to get one that's has everybody. We looked at them individually, but I want one. All right. As much as I think this guy talks too fast, look at this one first. All right, putting the code in the chat. Everybody log into this one. All right, while I'm waiting for everybody to log into this one, we'll look at the main point. Macromolecules are large, thus the word prefix macro. And in this case, they're still microscopic to you, but they when we can these are in terms of the cell membrane. Everything is in the cell is controlled by the cell membrane. So it's macro in terms of what can fit through the cell membrane. All right, they're necessary for life, um, and which is why we have to do some extra things sometimes to get them in the cell because the cell is keeping out big molecules, but we need these inside. Okay, the four basic ones are carbohydrates, lipids, proteins, and nucleic acids. Each are important to perform a wide variety of functions. So again, the worksheet that you have, the little chart that we made, tells you their functions. They are organic. Remember that word organic means that they contain carbon. Okay, these are molecules that contain carbon. Things that don't contain carbon, like water, um, are inorganic molecules. Okay, so in addition to carbon, they also have hydrogen, oxygen, nitrogen, sometimes phosphorus and sulfur and some other elements, but mostly they contain carbon, hydrogen, oxygen. You also have that on your chart. All right, we got everybody in yet? All right, collab board. Why are biological molecules considered organic? And I just gave you the answer to that. So this is your opportunity to repeat what you heard. Why do we call them organic? Why are they not inorganic? All right, and I'll make them visible in a minute, but got one person. Let's go, everybody. Why are they organic? Come on, folks. I should have 10 little blue squares up here on this board. Another minute, another minute. I got 6. I'm supposed to have 10. All right, let's see. Okay. Imani, you got too many. All right. All right. Junior, you did a, and folks, we don't have time for y'all to be looking up stuff, trying to get the encyclopedia version of the answer. All right. And technically, folks, it's just that, like the first two folks, Raymond and Jamarion, and who else? It's just because they have carbon. They are carbon based in Brooklyn and Zender and okay, who else? And Avion, Avonay. All right. Because they are carbon based. That's it. They are carbon based. All those other elements, some of them have them, some of them have all of them, some of them have don't have everything. They all mostly all have carbon, hydrogen, and oxygen. Some have nitrogen, some have sulfur, some have phosphate, but they all have carbon. All right. Um, so this is the picture is showing you a monomer of a glucose, which is a monosaccharide, but there's like 4 of them here. Okay, and what they're trying to show you here is that they are joined together by removing water. So you see the red down here at the bottom? We tend to say think of water as HOH instead of H2O. Think of it as HOH. One side is going to have the OH molecule, and the other side has a hydrogen that it's going to steal from the OH molecule. And those two, when they remove those two, the oxygen is going to link up or join hands like they have over here. And the water molecule is removed. Okay. So that's how and synthesis means that you're making a molecule. So these, we went from having 2 monosaccharides to having joined them together, and now we have a disaccharide, which is what your table sugar is. That's sugar that you sprinkle on your cereal or put in your tea. It's a disaccharide. Okay. And they call this dehydration synthesis. Okay. And right now, if you were in chemistry, you would have to know all of this, but right now we're not going to worry about this. Just know that 2 glucose monomers are put together by removing a water molecule through dehydration synthesis. Okay. There are different types of carbohydrates molecules, glucose. We have starch, glycogen, and cellulose. Those are your 3 different kinds of carbohydrates. Starch, remember, is stored glucose. We don't store glucose as glucose. We store it as starch and glycogen. And cellulose is only found in the cell wall of plants. All right. If we want to break that molecule apart, we add the water back. And then and when we add the water back, and think again of HOH, the hydrogen goes to one molecule and the OH goes to the other one, and it splits it back up. So this picture is showing you the reverse of the first one, where these first two are holding hands at the oxygen link. We add some water to it, and again, in the process, there's an enzyme involved in this too. One of the OH goes on one side and the hydrogen goes on the other. All right. We're gonna leave that one alone. Okay. All right. Okay.
Many important biological molecules are made of repeating subunits, called monomers. When many monomers join, the result is a polymer. For example, amino acid monomers join to form a protein polymer, and glucose monomers combine to form a complex carbohydrate polymer. Biological polymers form by dehydration synthesis reactions. As you can see here, each of the monomers in this reaction has a hydrogen, or H, and a hydroxyl, or OH group. In the course of the reaction, the hydrogen is removed from one monomer and the hydroxyl group from the other. The hydrogen and hydroxyl group combine to form water, and a bond links the two monomers. Hydrolysis is the opposite of a dehydration synthesis reaction. During a hydrolysis reaction, a polymer is reduced to its monomer subunits by the addition of water. In fact, the word hydrolysis literally means to break water. The hydroxyl group from a water molecule attaches to one monomer, and the remaining hydrogen attaches to the other monomer. In other words, water is used to break the bond holding monomers together. Let's do a quick recap. During dehydration synthesis, monomers join to form polymers, and water is released. The opposite happens during hydrolysis, where water is added to the reaction to break a polymer into monomers.
All right, you got a question, two questions to answer. Two glucose molecules linked to form a disaccharide, meaning they were a monosaccharide, now they're a disaccharide. Maltose is one of them, that's milk sugar. Releasing water by the process of what? If you think about they all through the summer they kept telling you drink some water so you don't get what? Dehydrated. Thank you so much. That was the correct answer. So think of it in terms, personalize things sometimes. And if they want you to drink water, you are hydrating yourself. Like all some they said, stay hydrated, so you don't get dehydrated. And that's because your cells, like osmosis in the pictures you had, are going to shrivel up. And then you're just going, and then they're not going to work well. And then you start feeling faint. All righty. Waiting for players. Let's wait for some players. I got 2 players, 3 players, 4 players. Oh, yeah, it didn't show the second question for me. I'm sorry about that. It went straight to this climb activity. All right, somebody's missing. Like y'all's variety. There you go. All right, here we go. Little dinosaur, huh? All right, I'm not gonna. Oh yeah, we can do that. There's a way to. All right, I'm gonna stop sharing this while y'all answer, then I'll pull it back up when you're finished. Remember, these questions are timed. All right, so Jamarian gets the gold star, got all the questions correct. I wonder if it let me go back and, okay, I'm gonna go back and look at these questions because okay. Yeah. And see, with the question two, for me, I'm looking at the slide, it skipped right over question two, so I have no idea what question two was. All right. And I'm just going to, oh, it won't let me start the game. Okay. Thank you. I just need one person. And I wonder if I can pause it. Thank you, Jamaria. Can I pause this? All right, question number one, most of you got okay. It was dehydration synthesis leads to a form where water and polymers. So you have two of the molecules. The second question, during the breakdown of the polymers, which of the following reaction takes place? If they're breaking down, hydration. Okay, because to break it up, you've got to add water. And most of you got that one wrong. All right, the next question, I can't pause this. The following reactions, yeah, it is going fast. How do you slow this thing down? Yes, concentration, which I don't know why you would know that. All right, which of the following polymers formed during dehydration? And that's going to be starch because the others are not polymers. Okay, so remember we had starch, glycogen, and cellulose. All right, so that was that. Okay. Yeah, the timer was getting me too. Okay. With the time that's left, I'm gonna let you just pull out your review sheets. Complete any worksheet that you might owe me, although most of you in this class, wonderful people that you are, don't owe me anything. And that's wonderful. Let me check. And that's miraculous in itself. You must be the only class where I'm not sitting at folks who owe me a whole bunch of work. Some of you owe one or two things, but you're not failing, and that's a good thing. So yes, two of you still owe me a transport. Two of you, one of you owe me a discussion, transport discussion question. And that's that. All right. So as far as the test is concerned, make sure you know what the it does cover everything. Know your scientific method in order. So let's take a look at that. Know the characteristics of living things. Ms. Grin. The questions are based around Ms. Grin. So know what Ms. Grin is. Um, oh, why did I do that? What is Miss Grin? That's a question. Somebody tell me, what is Miss Grin? And answer it for everybody. What do I mean by Ms. Grin? It's an acronym, and you need to know what each of the letters of that acronym represent. In general, what does it represent? Anybody know? That's like 7 questions on the test you need to know.
Life.
Go ahead. MRSGREN. Why do you need to remember Ms. Grin? Lovely. Raymond but Raymond, you sent it to me. Send it to everybody. And you put it in order too. There we go. Wonderful, wonderful. All right. So if you don't know, now you know. Okay. Ms. Grin stands for, and what it really is all the required activities to be considered a living thing. So if something does not do all 7 of these, then it is not a living thing. Okay, so you need to know why they need movement. What is considered movement? What is respiration? And remember, respiration is not breathing. Breathing is breathing. Breathing is the mechanical process of taking air in and letting air out. Respiration is actually a chemical process where you are combining or taking glucose and you're breaking it down. You add oxygen to it, and the oxygen oxidizes the glucose and breaks it down into carbon dioxide and water. But in the process, it releases energy. That is the important part about respiration. This is where you get your energy from, respiration. All right, sensitivity means, and it's not your emotion. I think that's one of the choices on the multiple. Has nothing to do with your emotions when you're being sensitive to people calling you names. It's where how an organism responds to its environment. Okay, all living things must grow. If it doesn't grow, for example, a rock cannot grow. You might be able to pick that rock up and move it from one place to another, but it didn't move by itself. So it's not a living thing. It can't grow. It can't move by itself. It doesn't respire, doesn't respond to its environment. Reproduction, all living things have to reproduce, make more of its own kind. And the cell theory, you need to know the cell theory that all cells come from pre-existing cells. Cells make cells. Okay, reproduction. All cells get rid of waste products, excretion. If you don't get rid of waste products, toxicity builds up and you die. Nutrition, they got to eat. Find a way to get nutrients inside. Plants make their own food. They make the glucose. The glucose is their food. Okay, that's how they get their energy. They make the food and then they use the glucose that they make to perform all of these life functions. All righty. So make sure you know that, like I said, that's 7 questions on the test right there. What else can we bother you with? Mm, like I said, study your organelles function, like mitochondria or your energy producing organelle, your um, is on the tip of my tongue. Ribosomes make the proteins. The nucleus has the DNA. That's where your genetic code is. Know all the major um, organelles. Let me pull up scientific method. All right. And just a heads up, scientific method isn't going to go away. It's just going to be something that you will cover in every science class that you go in. If you go to college and you write a dissertation, it uses the scientific method as the steps to your paper that you write. You have to state a problem. Okay. What is your problem? If you're writing a paper, they want you to state a problem. Then your thesis statement is the answer to your problem. And then you have to defend your thesis statement. So when you get to college, this is still the same format. You're going to have to show what you did to defend it. You're going to collect data while you're doing, and then you're going to come up with a conclusion as to why you feel this is the answer to the problem. Okay, so this is know the steps of the scientific method, and here they're explained to you right here. I believe this is a matching question on the test. I'm not sure. There's some matching questions on there. So this is the first unit on scientific method. So you might want to go back and take a look at those. Again, know all of your vocabulary, 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 vocabulary. All right. Like I said, I would stop. So we did transport, we did macromolecules, and no basic lab safety rules. There's some questions about lab safety. The general answer for that is always let the teacher know what's going to always do what the instructions say. Don't make up your own experiments. When you're in the lab, be dressed properly. We might come back to this later because there are some things later on when we do some of the labs that you need to know. All the parts of a graph. Some of you don't know how to graph, and you will have to graph your answers and then analyze your graph. All right, so you're going to have a title. You have an X and a Y axis. You need to have labels on it. And then everything has to have units. So what are you measuring these X and Y axis things with? Know the variables, independent and dependent variables are. So a variable is a factor that you can change, or the factor, the part of the experiment that does change. So if you change it, it's the independent variable. You manipulated it. And what happens when you manipulate it, the change you get when you manipulate it is the dependent variable. It depends on what you do. Okay. All right. So in this example, the light, if we want this plant to grow, we are going to vary how much light we give it. So we can change whether it gets a lot of light, a little bit of light. And then our dependent variable is, does it grow? How much does it grow? We can measure. Again, in an experiment, you have to be able to measure your results. So we can measure how much growth happened because you changed or regulated the amount of light. All right, what else can we talk about? All right, we didn't spend a lot of time talking about this, but in general, you need to know, 'cause I feel like you've done this before in another class, but you need to know the metric system is what scientists use. The unit of length is the meter, the unit of mass, if you're weighing something, is the gram. So we don't use pounds and we don't use inches, we use meters and grams, and we don't use ounces, we use liters for volume. Scientifically speaking, we measure everything in seconds. So you need meters per second. We don't use Fahrenheit, we use Celsius. The area is length times width, so that's going to be meters squared. And if we do the volume of the room, that's length times width times height, then that's going to be in cubic centimeters. Okay. Know your metric prefixes. And again, we just okay. So a kilo, that's the abbreviation for kilogram. A That's some M is for milliliter. This one is just meter. Oh, okay. Milliliter, sorry. See, I wasn't reading carefully. All right, and a millimeter. Yay. All right, I did all that to get here. All right, so you go over the lab safety rules. Remember, we did this activity here. So remember what you can and cannot do in the lab. What is safe, what is unsafe. There are some safety questions on the test. Okay, so read that out. All right, so I'm going to stop sharing. I'm going to give you time to do some reading for yourself, time to ask questions. Again, this is Tuesday, so I'll be on the Zoom from 1:35 to 2. If you have questions, if you need to fix something, is there something I need to open so that you can submit it? Now's the time to ask, because tomorrow is the last day that you can submit late work. Last day. So if you're trying to catch up on something, tomorrow's the day. We are going to, I believe microscopes are the next. No, that's the other class. So the next, we're actually gonna be talking about respiration, photosynthesis, cell energy. So if you want to get a head start, look at everything you can. That module is now open for you to look at. Cell energy. So we're going to keep talking about ATP in more detail. It's going to get slightly more complicated. We're going to add a little chemistry to this. Again, if there's something you just totally didn't understand, now's a good time to ask. Really? I know you have 6th period exams today. You can use this time to get a peek at your 6th period exam. Start preparing yourself for 6th period exam. Now to All right, if you don't have any questions for me, you can have an early lunch. And I'll see you on Friday. See you tomorrow. See you tomorrow. Have a good one.